Good afternoon, everyone. It's really an honor and a pleasure to once again introduce the George Khoury Memorial Lecture. A few biographical details about George. George was a graduate of Princeton and Harvard Medical Schools, came to the NIH in 1976 to serve as head of the virus tumor biology section in the Laboratory of Molecular Virology at the NCI. 4 years later, he became chief of that laboratory. 1981, he won the Arthur S. Fleming Award for Outstanding Government Service, and then in 1987, the year that he very sadly passed away at age 43, he was elected member of the National Academy of Sciences. Here's George with Peter Howley, who many of you know, and Malcolm Martin, who was my postdoc mentor. During his short career, George published over 140 papers deciphering mechanisms of eukaryotic transcription. He had a very impressive group of trainees and collaborators, some of whom are listed here. And correspondingly, over the last 20 or so years, this lecture has been given by a very august and illustrious group of speakers, including 2 Nobel laureates and a large number of National Academy of Science members. So it's fitting that this year, 2017, the George Khoury Lecture will be given by Patrick Moore, who serves as Pittsburgh Foundation Chair in Innovative Cancer Research at the University of Pittsburgh Cancer Institute. Pat is a native of Wyoming and Utah. He did his undergraduate work at Westminster College in Salt Lake City, master's degree at Stanford, MD at University of Utah, and an MPH at Berkeley. He also trained as an epidemiologist at the CDC and led research teams to control meningitis epidemics throughout the world. And Pat has some very interesting stories to tell about this period from his career, which I'm sure he'd be happy to share with you at the reception afterwards. In the mid-90s to the early 2000s, Patrick was on the faculty at Columbia University, and then in 2002 until the present, he's been serving as the professor of microbiology and molecular genetics at the University of Pittsburgh. Among many notable accomplishments, Patrick is co-discoverer, together with Yuan Chang, of KSHV and Merkel cell polyomavirus, and he's really been a pioneer in the study of how these and other viruses cause cancer. He's received a number of awards, which are listed here, and 5 years ago was elected to the National Academy of Sciences. So without any further delay, it's really a pleasure to introduce Dr. Patrick Moore as the 2017 George Khoury Lecture. His talk is entitled, Thinking About Cancer as an Infectious Disease.
Thank you, Eric. That was really nice. Thank you. Thank you, and I'm really tremendously excited to give this lecture and proud to be here. It's quite an honor for me, and I really appreciate it. So finding causes and also cures for cancer are probably the most complex and expensive intellectual endeavors that humans have done, I think. Certainly, it's over the last 50 years, it's been much more expensive. Many people have been involved in this effort than putting a man on the moon, Manhattan Project, many other things that have been done. And so there's always, over this past 50 years or so, been a question about, well, what are the role of viruses in cancer? And it's been very unclear. We know, obviously now, that viruses and other infectious agents are responsible for about 1 out of 5 cancers worldwide. And this is easily seen here. This is global CAN data. I'm sorry. This is global CAN data, WHO, and you can see that of all comers, that breast and lung among women and prostate and lung and colorectal cancer and so forth are the most common cancers that occur. Infectious cancers overall among this group are relatively small, probably less than 3% of all cancers. But let's take a look at a cancer registry in Kampala, Uganda, where we see a very different pattern of cancer. What we see is that cervical cancer is one of the most, if not more common than, it is more common here, I believe, than Kaposi sarcoma, but Kaposi sarcoma is quite common as well. KS is quite common among men, and overall, we see that 50% of cancers are caused by infection. Now, if you think about it, obviously, cancer registries in Burkina Faso, Laos, and other developing countries are far less likely to be sensitive for detecting patients. And so this 1 out of 5% of cancers worldwide is likely to be an underestimate. Furthermore, there's also a problem that many cancer registries don't record non-melanotic skin cancers, which Kaposi sarcoma is. And so there's an underestimate of the number of cases of patients who have cancers due to infection. And we can see that this is the standard list of the 7 viruses that cause cancer. And I'll be talking about these last 2 because I think that they have something interesting to tell us about how we can think about cancer, not just infectious cancers, but also non-infectious cancers. Now, Kaposi sarcoma was, of course, described by Moritz Kaposi in 1872, was the harbinger of the AIDS epidemic. And this graph is really remarkable. This is SEER data for young men in San Francisco County from 1975 to 2014. This is obviously the AIDS epidemic, where you can see that AIDS KS spiked up like this. Now, I was in medical school right here in the early 1980s. And when this happened, cancers do not occur as epidemics. They just don't occur as epidemics like this. And so for this epidemic of cancer to occur, this was really a remarkable instant in medicine. But obviously, we now know that this is caused by this virus, KSHV, that in the context of immunosuppressed patients with HIV has a high rate of developing this cancer. So this is my wife, Yuan Cheng, and Eric was kind enough to ask us both. He said, well, one of you has got to give the Khoury lecture, so who is it? And so Yuan and I did what we usually do, is we flip a coin and you lost. So I'm here, but she easily could be giving this lecture as well. And what we did back in 1993 was perform a subtractive PCR technique called representational difference analysis to isolate 2 small fragments of the genome of KSHV. Now, why is this interesting? Well, it turns out that KSHV is a large double-stranded DNA virus. It's like EBV. But, oops. It's like EBV, but what's different from the other human tumor viruses is that KSHV in its evolution has stolen genes from the host genome. And so when this was sequenced in 1996, it was quite remarkable to see that it had a viral cyclin, it had a B-flip, it has an OX2, it has a BCL2, all of these really interesting genes that, unlike EBRs or E1As or E6s and E7s, we could immediately assume that these genes for this virus were targeting specific cellular pathways that were already known. And so we had more of a systems biology approach to looking at how this virus might cause a tumor. And one thing that became quite clear is that many of these genes actually target the immune system. And to give you an overview of why we think this is occurring, I have to tell you a little bit about the life cycle of this virus. This, like other herpes viruses, exists as an episome in the nucleus of cells when it's latent, and it initiates replication through rolling circle replication to form viral particles, perhaps thousands in a cell that's initiating replication, and that will kill the host cell. Now, this is a process of lytic replication is dependent on a series of transcription factors that are encoded by the virus. Alternatively, the virus can exist as just a simple episome in cells, and it only divides with the host cell. Here, the virus uses its own DNA polymerase in order to initiate DNA replication. Here, it relies on the cellular DNA polymerase, and this is the reason why antiviral drugs like acyclovir work for herpes viruses, is because they specifically target this polymerase without targeting this polymerase. And so what that means is that we can inhibit lytic replication of herpes viruses, but we can't touch latent replication. Now, this is the form of the virus, the life cycle of the virus, that causes tumors. And the reason is, as you expect, is that if a tumor cell were undergoing lytic replication, it would die. It would form all of these virions, and it would initiate programmed cell death and die. Now, when we first found KSHV, the standard model for why viruses initiated tumorigenesis, why they had oncogenes, was the following, is that what they're doing is first driving the cell into S phase so that it has the proper environment in order to replicate its DNA. But if you have unscheduled G1S entry, then of course you have to inhibit p53 to prevent that cell from initiating cell death and dying before the virus has had an opportunity to replicate. So the point that I'm getting at is that the early views of why viruses had oncogenes were that these viruses were driving the cell to proliferate so that the environment was proper for the virus itself to replicate. Now, one of the first genes that S.J. Gao, who was born in China and came to the United States, immigrated to the United States, and was in our lab, one of the first genes that he looked at is a gene that was known as VIRF-K9. And we saw this gene, we thought, well, this is going to be an interferon inhibitory gene, and it was. This is an interferon reporter, and if we add interferon, or add increasing amounts of expression of K9 gene here, then we see that this reporter is turned off, and if we add interferon, the reporter turns on, it turns off. So it's inhibiting interferon. This is what we would expect a virus to do. It inhibits antiviral responses. But if we put the same gene into 293 cells, it turns out that it transforms them. So there's this yin and yang between antiviral activity and anti-tumor activity. And this was actually first noted by Taniguchi, at least formally described by Taniguchi in his description of IRF1, which is a pro-interferon, largely pro-interferon signaling transcription factor, and IRF2, which had counteracting activity and tended to turn off interferon signaling. IRF1 is a tumor suppressor protein. IRF2 tends to be a viral oncoprotein. So what we see is that there are features of innate immunity, and we would anticipate that if a cell wanted to prevent a virus from replicating, it would inhibit the cell cycle, it would induce apoptosis or some form of programmed cell death within an infected cell, and then it would marshal the resources to initiate an adaptive immune response. Well, these are exactly the same features of tumor suppressor proteins: to initiate cell cycle arrest, initiate programmed, cell death, and activate an adaptive immune response. Now, getting back to the replication, this is the reason why I want to tell you this story, is getting back to that story about how we thought viruses caused cancer, is the idea that viruses would infect a G0 cell and drive the cell into the S phase, drive the cell into cycling, cell cycle, initiate S phase so that it could multiply its own DNA, and that would create viral replication and cell lysis. Now, if we look at KSHV, KSHV is one of the few viruses where we have a very clear pattern of latency that we can manipulate in the laboratory. EBV is another one. And if we take a look at the gene expression of the major locus that is involved with transforming cells, here we can do northern blots. That's how old this data is. These are northern blots. And what we see is that if we induce replication of the virus or inhibit replication of the virus, there's really no change in gene expression. So these genes are not being induced during the period when we would anticipate these oncogenes should be expressed in order to promote replication. So is there something else going on? We think it's the following, that instead of an arrested cell, if a virus infects a cycling cell, it can initiate innate immune signaling pathways, particularly that end up activating P53 and RB, but many other signaling pathways as well, to arrest or promote a pro-apoptotic phenotype. And so in order for this virus to survive, it has to express these viral oncoproteins to avoid this fate. So that suggests cell death during viral infection is actually a cellular response to viral infection rather than being driven by virus infection itself. If we look at some of these genes that I've told you about, I won't go through them in any detail and tell you what they are, but you may know just by their abbreviations what they are and what they do. All of them look like good bona fide genes that should be involved in regulating immunity, and particularly innate immunity. But on the other hand, so they look like they're innate immune evasion genes, but on the other hand, they're all transforming as well. So it looks like we have a yin and a yang between innate immunity and tumor suppression that is revealed by tumor viruses. And of course, all of these papers, many, many papers you can see every week in immunology, Nature Science, so forth, with P53, P10, STING, CGAS, so forth, suggesting that these either innate immune signaling pathways or these tumor suppressor pathways are active in either tumor suppression or innate immunity. So there's this duality. Why is there this duality? And what we suppose is the following. This is a picture, and I'm sorry you can't see this, but you can imagine that this is a phylogenetic clade, and down here is a unicellular organism. We're somewhere up in here. I'm not really certain where we are. But what you can imagine is that this is the evolution of P53. So P53 existed as a functional protein in unicellular organisms. What is it doing? It appears that it's probably part of an innate immune signaling defense pathway in these colonial organisms that prevents infection from spreading from neighbor to neighbor to neighbor. And that as we evolved into multicellular metazoan organisms, then these innate immune signaling genes, like P53, were co-opted to control tumorigenesis as well. And so we have two things going on, and we haven't lost the ability to control viral infections. It's just that many of these same pathways are acting also in tumorigenesis, preventing tumorigenesis as well as viral infection. Now, we can stand this equation on its head as well, right? So if we have a tumor which has a series of mutations in various tumor suppressor genes that happen to knock out innate immunity, then those tumor cells should be more susceptible to viral infection. And in talking to my friends who are in viral oncolytic therapy, that's precisely what is happening, is that those tumors that are responsive to oncolytic viruses like herpes viruses or pox viruses, they're most active only in those tumors in which particularly interferon signaling pathways are unresponsive or are knocked out. So I would like to switch gears now and tell you a little bit about MCV, which I think is another interesting virus. And I have to apologize. Unfortunately, your Wi-Fi network keeps on wanting to talk to us, and I don't really think we need to talk to it right now. This is a cancer, skin cancer, that Eric Engels here at NCI actually first noticed was increased about 11-fold among AIDS patients. And that seemed to you and I to be a likely candidate to be a tumor that had a viral etiology. So we worked for about 7 years to develop a method to directly sequence RNAs from tumors. And this culminated in digital transcriptome subtraction that was spearheaded by Wei-Cheng Feng, who immigrated from China, and Masashuda, who immigrated from Japan, to perform deep sequencing on these tumors, and then virtually subtract out, just as we subtract out sequences for KSHV, now we're virtually subtracting out the RNAs, leaving us a pool of candidate sequences down here. It turns out that 2 of these sequences belong to a virus that was highly related to the virus that George Khoury made so much progress on, and that's SV40. It's a polyomavirus, and it turns out to be the only polyomavirus that is clearly established to cause human cancer. This is a small DNA tumor virus. It is present in about 80% of Merkel cell carcinomas, and it's clonally integrated into Merkel cells. It only has 2 genes, an early gene that is alternatively spliced in various ways to form various proteins that I'll tell you a little bit about, and a late gene here that also has different isoforms that form the capsid, package this capsid, and transmit the virus. So what have we learned from this virus? Well, first of all, here are the 2 main early proteins that the virus expresses. One is large T, which is this spliced protein here. It's actually a helicase. And what it does is it sits on the viral origin, assembles as a double-headed hexameric helicase, opens up the DNA, and draws the cellular DNA replication machinery to that site to initiate replication of the viral genome. The small t is a little bit more ambiguous what it does, but the small t, I'll tell you, we believe that it binds to E3 ligases as well as to protein phosphatases, particularly PP2A. And both of these proteins are expressed in some tumors, as you can see here. Other tumors, the small t is more predominant, but I believe that it's likely that there is some degree of small t expression here, or large T expression here, and that's also driving the tumor. So this is a diagram of large T. Here's the helicase domain. The tumor suppressor targeting domains are here in the N-terminus. It turns out when Massa and Feng started sequencing this virus, all of these genes had mutations in them that truncated the helicase domain here. And so all of the tumor-specific antigens are these rump proteins that only have the tumor suppressor targeting domains in them. The helicase domain is absent. So we have this idea now that these are the molecular steps of the genesis of Merkel cell carcinoma, is that infection is near ubiquitous. Most of us in this room are infected. We were probably infected as children. But at some stage, there is, perhaps due to loss of immune surveillance, perhaps due to other reasons, an initiation of active viral replication. And how do we know that that's the case? This is work that we performed together with Chris Buck here at NIH. And if we take a look at the antibody titers against Merkel cell carcinoma among patients who are positive, there's sky-high antibody titers against the capsid proteins here, much higher than we see in MCV-negative patients or in blood donors on average. But in the tumors themselves, the capsid proteins are not expressed. They're not expressed. So this is a bit of a dilemma. But what we think then happens is, due to this initiation of uncontrolled DNA replication, you have defective replication intermediates that then, through non-homologous end joining, get integrated into the host cell, and that's not so bad either in terms of tumor genesis. And the reason is because, as I mentioned, the large T antigen binds to its origin and initiates replication. But if it does that, it's going to run into a replication fork from the human chromosome, and that's probably going to cause fragmentation and kill the cell. However, if there's a secondary mutation, like the ones I've talked about, that eliminate this helicase function, then you have unopposed expression of the viral oncoprotein here that can lead to Merkel cell carcinoma. Now, what I've told you now is, the interesting thing about this is part of our flora. You can develop cancer by having your flora being mutated without driver mutations in host cells, apparently. Now, the second thing that I think is quite interesting is new work that's come out from Hyun-Jin Kwon in our lab, who's from Korea. And what she's found is the following. Now, remember, I told you that this is an infection and that this infection is lifelong. But there's no definition of latency for polyomaviruses. Now, if we go back, this is a slide that I used to use in my teaching when I taught epidemiology 10, 15 years ago. And I'm certain that all of you know these terms, the basic reproductive rate of transmission of an infectious disease, and then you can have changes to that so you have an effective reproductive replication rate for a pathogen. And if this number is greater than 1, then you'll have an outbreak of disease. If it's less than 1, then the pathogen will die out in population. We can use the same logic to apply that to individual organism. If I'm infected with MCV and the replication rate of that virus is anything other than 1.00000, then if it's above 1, I'm going to turn into about 94, maybe 95 kilograms of MCV DNA within 5 or 10 years. That's just simple logic. That's what's going to happen. Or the virus is going to die off, and I will no longer be infected with this virus. And so how does a virus with just 2 genes so closely and carefully define its own replication so that it is not either turning me into a big ball of MCV DNA or being lost? Well, first we need to define viral latency. And when we define viral latency, we've thought about this a bit, and I think that there's not any good definition that you can have other than the following, that the viral genome during infection does not replicate independently of the host genome. Obviously, that also implies that it's not producing capsid particles or anything else from any virus that is latent. And latency is well defined for herpes viruses, and it's also well defined for retroviruses. Many of you here are working on these problems. But it's completely undefined for MCV and other small DNA viruses, or for RNA viruses. And yet we know these are lifelong infections. So how are these viruses maintaining themselves? And so this is Hyun-Jin, and what she did was she began looking at E3 ligases. What she realized is that SEF-FEW7, which is a ubiquitin E3 ligase, SEF-E3 ligase, targets the T antigen, initiates ubiquitination of the large T antigen, and degrades it spontaneously. However, small t's function appears to bind to FBW7, displace it, and to maintain higher levels of T antigen. And so small t here is acting as an accessory replication protein by increasing levels of large T in cells. And this is just a simple immunoprecipitation where you can see that LT is binding to FBW7, and here, if we add small t, we eliminate this interaction. If we mutate the FBW7 binding site, then it goes away. Well, so this implies that there's going to be a specific sites on the large T that are targeted by FBW7. And it turns out that large T is a densely phosphorylated phosphoprotein, has about 82 sites that are potential phosphorylation sites. And so she identified 15 sites that were either likely FBW7 or another E3 ligase, SEF E3 ligase skip2, or beta TRCP phosphorylation sites. And she mutated each one of these sites, put an alanine at each one of these threonines or serines so that the protein could no longer be phosphorylated. Now, when she did this, what happened was the large T protein, which again is the major replication protein for this virus, increased only with this mutation at 147, at 220, or 239. And I won't go through all the details on this, but it turns out that this is the beta TRCP binding site, this is the FBW7 binding site, and this binds skip2. So if we introduce these mutations into the virus and then put this back into cells, we can get the virus to replicate. And if we mutate the origin here, then we can get a replication-dead virus as a control. If we do this, here's the wild type virion. This is full genome that is put in to 293 cells. What we see here is some low level of replication. If we put a mutation that blocks origin binding, it drops down. It turns out that if we mutate this site at 147, it also decreases replication. But now something interesting happens. If we start to mutate at this site, which binds FBW7, or S239, which binds SCIP2, and we actually have increased replication over wild type. We're making a more replication competent, a better virus than exists in nature. And we can see that the proteins are expressed better when we mutate this. We can see that the viral DNA is bound better. And furthermore, whereas this virus is utterly dead for transmission, if we use a trans-well system where we can transmit this virus, this is the wild type virus, we see no transmission here. But once we start mutating these E3 ligase recognition sites, suddenly we can initiate replication. The red here is the VP1 protein, the yellow is, the green is the LT protein, and we can start to see transmission of this virus. Not robust, but here this is how we believe this virus is maintaining itself as a latent virus that needs to persist for years and years and years, and this is precisely why it's a risk factor for Merkel cell carcinoma. We know that also if we add antibody to it, we can neutralize the transmission. Here is going from the wild type virus to the mutant virus up here, but if we add antibody, we can knock it back down again. So that's part of transmission. And I want to add an interesting finding is that it turns out that mTOR kinase inhibitors, AKT PI3 kinase mTOR inhibitors, this signaling pathway will turn off beta-TRCP and SCIP2 expression. And all of these increase LT expression. So serum starvation also will turn off mTOR. And so here's wild type virus. If we starve the cells that have the viral plasmid in it, we'll start to see expression. If we add an mTOR inhibitor, again, we'll start to see transmission here. And this truly is lytic replication, where these cells are initiating programmed cell death. So what I want to summarize for you is the following, is the idea that even this small genome uses a mechanism that we call protein-mediated viral latency, where its replication protein has evolved to be constrained in its expression under most circumstances. But under very specific circumstances, where the virus would find it to be advantageous for it to replicate, particularly through these E3 ligase signaling pathways, if these are turned off, then we can see that LT protein will build up, bind to the origin, and initiate viral replication. It's a very simple mechanism, but we believe that this is the reason why this virus can persist lifelong. Do other viruses do it? Well, there's another virus. This is a virus that Chris Buck found, HPYV7. It causes a really unpleasant skin rash in transplant patients. And it turns out that if we inhibit mTOR signaling, either with PP242 or serum starvation, again, we can see activation of viral replication. How about RNA viruses? Well, a group in Japan, or a group in China, has shown that the JFH1 strain of hepatitis C virus, if you knock out FBW7 in these cells, it will increase virus replication. And the reason is because there's FBW7 binding sites in the NS5B protein of hepatitis C virus. And this is the RNA-dependent RNA polymerase. So that's very strange. Again, it appears that hepatitis C virus is limiting its own replication using these signaling pathways. Now, in terms of treatments, herpes viruses, again, you can treat herpes viruses with acyclovir, and about 5-7 days, you'll eliminate this active replication. You won't touch latent replication, but you'll inhibit active replication. If we use neuraminidase inhibitors for influenza A, about 5 days will prevent infection with this. But in patients who are treated with highly active protease inhibitors for hepatitis C virus, it takes 3 to 6 months to eliminate all hepatitis C virus from their liver, suggesting that much of the virus is sitting there for prolonged periods of time and is not actively replicating. So this may not be true latency, but it is probably a form of persistence that the virus uses and actually may have some important implications for drug therapies. Now, I don't want to imply that this is the only mechanism that these small viruses use to maintain latency, but I think that it is likely to be an important one, at least for some viruses. There are other mechanisms, for instance, mRNAs that viruses encode that target their own replication proteins. They can inhibit their own replication protein promoters and so forth. So what have we learned from these viruses that I think can inform you at least a little bit, even if you don't care about tumor viruses at all, but you do care about cancer? There are some things that this work and the work of my colleagues can tell you that I think is quite important. First, there are direct benefits from tumor virology. As I mentioned, 1 out of 5, at least, cancers, I believe, worldwide are caused by infection, and we really should be targeting those cancers more actively, as we've seen with the development of the HPV vaccine. This is something that we can initiate quite easily to change the course of human cancer. There's an interplay between immunity and tumor suppression that I don't think everybody is quite clued into, but I think it's quite important. I wanted to mention that we've learned something about viral flora, that mutations of viral flora can contribute to carcinogenesis. And then also these viruses have provided novel ways to look at persistence and latency for these viruses and how that can contribute to carcinogenesis. I want to thank all of our laboratory, particularly Xiaojiang Gao and Ronit Reed were alumni from our labs. I've mentioned other people in the lab who've contributed, and our colleagues, Chris Buck, Bob Yarshwin, and others. I also want to point this out, and I'm not being trivial when I say this, is that you are in a really unique place, the NIH. I don't think that there's any place like it in the world, and that the NIH funds our research. We wouldn't be able to do this research without it. So what I'd like to do is say thank you. Thank all of you and all of the administrators. Sometimes I don't get along with them, but all of the other people at NIH for what they do. The scientific enterprise in the United States wouldn't be anything like this if this didn't exist. So thank you very much, and I appreciate your patience, and I'll be happy to take any questions that you have.
So you showed high rates of KS in Africa. How much of that is associated with HIV infection? Is that all
most of it, no. It's not all associated with HIV infection at all. Prior to the AIDS epidemic, Kaposi sarcoma and the limited number of surveys that were done in colonial Africa was generally the third most common cancer in Africa. So there are areas where it's highly prevalent. We know that The infection rate, infection prevalence in Africa, depending on the country and depending on the part of East or West Africa, can be anywhere between 30 and 70% in the general adult population. So obviously when the AIDS epidemic came in, then we saw something that was analogous to what we saw for the San Francisco SEER data, that there was an intensification of an existing epidemic that was already there.
I have a broad question about if you could comment on your thoughts on, in the context of immune suppression, why only certain viral infections seem to flourish in those environments and we don't see breast cancer, prostate cancer, other things, why certain viral infections flourish in the context of immune suppression.
Right, so particularly if I can't say much about viral infections in general, but in terms of cancer, what we see is that, and this is of course well known, is that both in transplant patients and in AIDS patients, that the cancers that have an increased rate over the general population are those that have a viral etiology. They're expressing foreign antigens, and so that's not surprising. What is a little bit surprising is things like stomach cancer also can be elevated among transplant patients. And that's a cancer that we would expect to be a hit and run phenomena with Helicobacter, but perhaps it's EBV that's contributing a role to it. But it's a bit of a mystery to us as to how that could be occurring. So the bottom line is that those cancers which have a foreign antigen that's driving the replication of the cancer, it's not very mysterious that those cancers would be under immune surveillance. And if you lose immunity to a pathogen, then you're at risk for developing cervical cancer, Kaposi sarcoma, Merkel cell carcinoma, and so forth.
You mentioned new tumor virus created new signs. Do you have any idea the possible new tumor virus come out in the next few years?
Oh, yeah. Well, that was a big surprise. What we thought that when we found MCV is that this would be the first of the next 10 or 15 tumor viruses. So the field is in a bit of a quandary right now. Is the 8 viruses that we know cause cancer, 7 viruses that we know cause cancer, plus BK, which probably causes cancer as well, BKV. Is that it? Is that the entire list? Or are there additional viruses or new forms that we haven't really worked out and haven't seen yet that are responsible for forms of cancer? I hate to say endogenous retroviruses because saying that is quite controversial, but I mean, that would be a viral infection that would not necessarily be under immune surveillance because you're born with a particular repertoire of endogenous retroviral proteins, and you probably wouldn't have a potent immune response to them. So that would be a tumor that was caused by an endogenous retrovirus would behave very differently from the tumors that I've described. So those are just examples. I'm not saying that those are real examples of additional cancer viruses, but we also have to pose the possibility that maybe we've done such a good job that there are no more cancer viruses. I don't think that that's the case.
Yes, not quite a long time ago when I was a resident in pathology, we thought that Merkel cell was essentially small cell carcinoma. And if you look in some of the older textbooks, that's what we pretty much called it. Now, subsequently, the Merkel carcinoma, as I understand it, there's 2 types though. There's one that's virally induced and one that's mutationally induced. And my understanding is the one that's virally induced actually can undergo spontaneous regression. And the reason that I would think that this is significant in terms of the new immunotherapies for cancer, Merkel cell has been kind of a poster child for PD-1 and CTLA-4, one of the 2, I forgot which one, but you can't tell if a Merkel cell, there's several case reports of Merkel cell and spontaneous regression. It's undisputable. But so then when you add it with the new immunotherapies for cancer, such as PD1 or CTLA4, you can't tell if it's spontaneous regression or if these agents are actually having an effect. Do you have any comments on that?
Yeah, so checkpoint inhibitors have been really remarkable for Merkel cell. And as you correctly mentioned, there are Merkel cell carcinomas. About 80% of them are, have the MCV in them, are caused by MCV. Another 20% are not. But if you take a look at those 20% of MCV negative Merkel cells, virtually all of them have an extremely high UV-induced mutation patterns. UV exposure is a risk factor for Merkel cell carcinoma. So presumably, either the virus can infect the precursor cell and drive proliferation of that precursor cell, or you can have 10,000 random mutations in your genome and ultimately wind up with the same pathways being abrogated as one virus. And so the one that has the virus in it obviously is expressing a foreign antigen that presumably would be benefit from checkpoint inhibitor therapies. And it turns out that both forms are equally suppressed, both the virus positive and the virus negative. And it's presumed that among the virus positive Merkel cell carcinomas, the reason is because there's this high rate of neoantigen presentation from the UV mutations that are present in the MCV negative tumors. You mentioned spontaneous remission, and that's actually probably a very good additional measure that there is a foreign antigen, perhaps due to viral infection, but an antigen that also is present in the tumor that is a fundamental component of the tumor process, and that in those lucky few that have, say, a reconstitution of a CTL immunity against that tumor antigen, then their tumor spontaneously clears, which is a rare event. And that's true. That happens with Merkel cell carcinoma, and it may also be an indicator of other cancers that have a viral etiology, whatever those cancers might be.
Okay, thank you.
All right, let's thank Patrick and Steve.
Thank you.